Hi everyone, my name is Wei Ting and I'm from the University of Sydney. So today I'm going to talk about the in vitro comparison between the originator and generic fluticasone salmeterol dry powder inhalers. So dry powder inhalers or DPIs are one type of devices commonly used for pulmonary delivery. They are widely used in the management of chronic respiratory conditions, for example asthma and chronic obstructive pulmonary disease. One of the leading products in the market is salmeterol. It delivers fluticasone propionate with salmeterol cinefolate. Its manufacturer is GSK. The first generic salmeterol in USA is Whistler Inhale. It has gained the approval in the beginning of 2019. Its manufacturer is Maitland. Interestingly, in India, there are different alternatives of generic salmeterol. Two of the largest companies, Lupin and Cipla, have their own generic products. They are EasyFlow Lupihaler and ZeroFlow Repholizer respectively. They have been available in India for many years already. However, they have not made themselves available in any Western countries. At the same time, there are not any published in vitro and in vivo data about them at all. Therefore, the aim of our study is to compare the dispersion performance among salmeterol and the two Indian generic products. The dose that we're using is 500 micrograms of fluticasone propionate with 50 micrograms of salmeterol. Two types of pharmacophobia in vitro aerosol performance tests were done. They are the delivered dose uniformity runs and the aerodynamic particle size distribution test. All the dispersion were done in a controlled condition at 50% relative humidity and 22 degrees Celsius. The flow rate was set at a 4 kpm pressure drop of each inhaler. The dispersion duration is the time allowing 4 liters of air to pass through the inhalers. For the delivered dose uniformity runs, it is to test how reproducible the doses are. The equipment that we're using is the dosage unit sampling apparatus. The inhaler connects to this side of the dosor, while the other end connects to the valve and a pump to control the flow rate and to generate the flow. After dispersion, we chemically quantified the amount of drugs per dose. For the aerodynamic particle size distribution, the aim is to separate the drug particles according to the aerodynamic sizes. The equipment that we're using is the next generation impactor. The inhaler connects to this side of the induction port, or the other end of the impacted body connects to a valve and a pump. The right angle induction port is to mimic human flow for impaction. The pre-separator here is to capture all the large carrier particles to avoid overloading the first stage of the impactor. The principle behind is that large particles have a higher inertia, therefore it cannot turn around the corners and it tends to stay in the upper stages. Small particles has a lower inertia, therefore it's easier for them to turn around the corners and go to the lower stages in the impactor. After dispersion, we chemically quantify the amounts of drugs in each size fraction. From the dosor runs, we calculate the emitted fraction, which is the emitted dose with respect to the labeled dose. This shows us how much comes out of the inhalers. Salmeterol emitted approximately 100% of the doses, which means that all of the dose came out of the inhalers. This is significantly higher than the two generic products. These generic products emitted approximately 60% of fluticasone and 80% of salmeterol. In other words, 40% of fluticasone and 20% of salmeterol is trapped inside the inhalers and could not come out. It means that the patient using the Indian generic products are likely to get less dose from each inhalation. For the aerodynamic particle size distribution test, two parameters were reported here. The first one is the mass median aerodynamic diameter, or MMAD. It is the aerodynamic diameter that divides the particle size distribution by half. With a larger MMAD means that the overall particle size is also larger. The ideal MMAD should lie between 0.5 to 5 micron, and it affects the location of lung deposition of the particles. The second parameter is the fine particle fraction, or FPF. It is generally referred as particles which is less than 5 micron of aerodynamic diameter. With a higher FPF, which means that there are more particles deposited in the deep lungs, and therefore the potential therapeutic effect is likely to be stronger. For the MMAD of fluticasone, serotonin is significantly smaller than the two generics at around 3 microns. The two generics have the MMAD of fluticasone at around 4 to 4.5 microns. For salmeterol, the three inhalers having a comparable MMAD at around 3 to 3.5 microns. So for particles that have MMAD at around 4.5 micron, for example, fluticasone from the generic inhalers, they are more likely to deposit in the central airways. For particles having a MMAD at around 3 microns, for example, fluticasone generated from steroid inhaler and also salmeterol from all three inhalers, they are more likely to deposit in the peripheral airways. For the fine particle fraction of fluticasone, steroid have a comparable FPF to steroid inhaler. Loop inhaler has the lowest FPF among the three. For salmeterol synthorate, all three inhalers have a comparable FPF. This means that in terms of the bronchodilation effect of salmeterol, these three inhalers are likely to be similar. However, for the anti-inflammatory effect of fluticasone, loop inhaler is likely to be the weakest among the three. However, since the in vivo correlation has not yet been established, whether the differences in vitro FPF has any impact on the therapeutic effect is still unclear. To conclude my presentation, steroid inhaler performed differently to the two generics in vitro. With the significant differences of the in vitro results, we still need more in vivo data to confirm their clinical significance. These are my references, and this is the end of my presentation. Thank you so much.